Hey guys, I'm so sorry that I couldn't be there today, but I did want to put your mind at ease and touch on a couple of the main ideas over organic molecules. So if you want to follow along with your notes, that might be a great idea. We're going to get started with our friends from last time, the carbohydrates. Hopefully, as we're thinking about carbs, we're thinking about short-term energy and some of those structural mechanisms. This should remind us that we're looking at monomers of monosaccharides, which can join through dehydration synthesis to form disaccharides, and we can add more and more subunits to ultimately form polysaccharides. Again, here's our dehydration synthesis reaction, joining things together by removing those waters. And remember that when we form these covalent bonds in carbohydrates, they're called glycosidic linkages. Remember, each of our organic molecules will have a different name for their covalent bonds, but carbs have glycosidic linkages. Here we're reminded of our two main examples of energy storage polysaccharides. That is starch. Remember, this is how plants store their energy in the form of carbohydrates. We also see glycogen, and this is how animals store their energy in the form of polysaccharides. We can tell that these are made up of these chains, sometimes branching, sometimes not, of monosaccharides all assembled, again, through dehydration synthesis. Remember, though, that not all carbs are for energy. Some can also have structural functions. A great example of a structural polysaccharide is cellulose. Yes, it is still a bunch of glucoses put together, but instead of the alpha linkage that we saw in starch, now we see a beta linkage. And these beta linkages are really, really strong and allow these strands to cross-link, which is why cellulose is such a great structural polysaccharide, forming the plant cell walls. Whereas the starch was all about the energy storage. The alpha linkages in starch make them much easier to break apart, which is why it's perfect for energy use. Remember, structure determines function. Another important structural polysaccharide is chitin. Remember, chitin is found in the exoskeletons of insects along with the cell walls of fungi. Hopefully thinking about starch, glycogen, cellulose, and chitin kind of brings your memory back to the main ideas of the energy and structural functions of carbohydrates. The next biological molecule we're going to look at is lipid. And here we can see a triglyceride or triacylglycerol. This is a great example of a lipid. And remember, the one thing all lipids have in common is that they're all hydrophobic or nonpolar. We can see this glycerol backbone has been joined to 3 fatty acid chains, and that building has been carried out by dehydration synthesis reactions at each of these 3 points. Remember, in carbohydrates, their covalent bonds were called glycosidic linkages. Also remember, in lipids, then, their covalent bonds are called ester linkages. This looks like a giant letter E, which is kind of convenient because their bond name also starts with the letter E. This diagram reminds us why these lipids, these fatty acid chains, are nonpolar or hydrophobic, and that is because they're primarily composed of hydrocarbons. Hydrocarbons, lots of C's and H's, and they share electrons evenly. When they share electrons evenly, that's a nonpolar covalent bond. And remember, nonpolar is the same as hydrophobic because nonpolar things do not get along well with polar things, like water. Here we're reminded that fats can be classified as either saturated or unsaturated, and that's going to be determined by the presence or absence of double bonds. So in the saturated fat, notice that there are no double bonds. in the saturated fat, which means that these form straight chains. The straight chains can pack together, making saturated fats typically solid at room temperature. And also, when they stack, they can form plaque, and that means that these can predispose you to cardiovascular issues. Over on the unsaturated fat side, we can see this is an unsaturated fatty acid chain due to the presence of the double bond. The double bond is going to result in bending or a kink in that tail. This bending means that the unsaturated fats do not pack together as tightly, making them liquid at room temperature. These are typically more your plant-based fats, whereas the saturated are more the animal-based, like we find in meat. Don't forget that phospholipids are another great example of a lipid. We still have that glycerol backbone, but instead of having 3 fatty acid tails, phospholipids only have 2. Notice I've got one saturated tail here, and they've pictured one unsaturated or bent tail here because of that double bond. So there are only 2, but I'm wondering what's going on at the third carbon. Well, in a phospholipid, the third carbon doesn't have a fatty acid chain. Instead, it's got a phosphate group on it, and that's what makes it a phospholipid. These are going to be a huge component in our cell membrane, forming the phospholipid bilayer. While the tails are hydrophobic due to the hydrocarbon chains, the heads are actually hydrophilic. They've got some greedy electron hog oxygens up here, so there are going to be some uneven pulls, which makes this polar or hydrophilic. Finally, for lipids, don't forget about steroids. They look a little different, but they are still nonpolar. They're going to be made up of these 3 hexagons and a pentagon with this variable chain. Different structures mean that these steroids can all have different functions. So there's our carbon lipid recap, and now let's dive in to some of our new material, the proteins. Remember that proteins are made up of monomers called amino acids, and these are going to be characterized by a central or alpha carbon. And hanging off that carbon, we see an amino group along with a carboxyl group, also a hydrogen. And there are 20 different amino acids. What makes them all different is this variable R group, or the side chain. There are a bunch of different things that this R group could be, 20 different options to make 20 different amino acids. Here are those options that you do not have to memorize, but I did want you to see that some of the R groups, shown in yellow, are hydrophobic or nonpolar. And we can tell that those are nonpolar side chains because they really don't have any greedy old electron hogs. They're pretty much just C's and H's, sharing evenly. On the other hand, these green R groups do tend to have some greedy old electron hogs, so these are going to have polar characteristics. These are amino acids that will interact well with water. Also, sometimes we can see acidic options for R groups, and there's some basic options as well. But each of these 20 amino acids simply differ in terms of that side chain, that variable R group. Here we're reminded that, again, we see a large macromolecule put together by dehydration synthesis. Here we can see a couple of amino acids, and we're going to add a new one by removing that water. Dehydration synthesis will result in a covalent bond between these subunit amino acids. Ultimately, we're going to form a polypeptide chain, and their fancy name for these covalent bonds is a peptide bond. Notice that these amino acids are lined up with the amino group on one side, that's called the N-terminus, and we see the carboxyl group on the far other side, and that's the C-terminus. And this is going to allow for each amino acid to have its N and C and N and C and N and C, all lined up so that a simple water can be removed to result in that peptide bond. Proteins can be really complex in terms of their three-dimensional structure. So we can describe 4 total levels of structure. The first is called the primary structure, and this is simply the order that their amino acids goes in. And this is determined based on our genetic code. We read our DNA so that we know which order to put all of these amino acids in. And each of these is attached to its neighbor through peptide bonds. So peptide bonds help stabilize the primary structure of a protein. Proteins can also undergo 2 characteristic levels of their secondary structure. Sometimes when hydrogen bonds stabilize this sort of curlicue, we call that an alpha helix. Sometimes it stabilizes a more accordion-like pattern, and those are called beta-pleated sheets. But either way, secondary structure is a result of hydrogen bonding along the backbone of these amino acid chains. As these chains fold over on themselves, they're going to have R group interactions, meaning that those variable side chains can interact with one another and help form even more complex folding patterns. There's several options. Sometimes we see some hydrogen bonding, sometimes we see some van der Waals interactions or ionic bonds. These are just R group interactions though, and the most significant, the most stabilizing of these is a disulfide bridge. This is where one side chain with a sulfur bonds with another side chain with a sulfur, and this is really going to help stabilize any of those folds and twists in the polypeptide chain. Oftentimes, we'll find that proteins are made of multiple polypeptide chains, and when they glue together, we call that quaternary structure. That's the 4th and final most complex. We can see a great example here. Hemoglobin has 4 polypeptide subunits, 2 alpha and 2 beta chains, and as they glue together, we're going to call that the quaternary structure. Primary structure was stabilized by peptide bonds. Secondary structure is stabilized by hydrogen bonds. Tertiary structure is all about those R group interactions, like the disulfide bridges. And when multiple subunits get together, that's the quaternary structure. Our final organic molecule is the nucleic acid, and we're going to spend a lot more time on this when we deal with molecular genetics. But for now, we need to remember that we read our genetic instructions. DNA is converted to RNA, which is then read by ribosomes to build our polypeptide chain. So it's really our instruction manual on how to make proteins. DNA is going to be the double-stranded nucleic acid. And in the double strands, we're going to see that guanine base pairs with cytosine, while adenine base pairs with thymine. Keep in mind, though, RNA is just a single-stranded molecule. Nucleic acids are polymers made up of monomers called nucleotides, and each nucleotide has subunits of its own. The nucleotide is made up of the sugar, the phosphate, and the nitrogen base. Keep in mind that DNA has deoxyribose, whereas RNA simply has ribose. The options for the nitrogen bases can be pyrimidines, which are cytosine, thymine, and uracil, or purines, which are adenine and guanine. As these polymers are assembled, we're going to see bonding down this backbone of the molecule, and that's going to be accomplished again by dehydration synthesis. We're going to see these adjacent nucleotides attached to one another when a water molecule is removed, and that's going to result in a special covalent bond, this time called a phosphodiester bond. We had glycosidic linkages, we had ester linkages, peptide bonds, and now phosphodiester bonds. Hopefully this has been a great reminder of all the key characteristics and functions of each of the 4 major organic molecules: carbs, lipids, proteins, and nucleic acids.